proteins. Okay. So proteins also, we hear about proteins, right? Proteins means we are in our daily diet also we are using different protein rich food items, right? Yes, the most abundant biomolecules in the biological system are proteins. See, protein content is present in fish, meat, dairy, cheese, cereals, peanuts, and seafood. See, this protein is very important for our health, very important for a living organism. So many of our dietaries contains these protein content. We have to include these protein rich food in our meals. Understood? Yes. So they are found in every regions of the body and serve as a foundation of the structure and activities of life. That is very important. They are also essential for body growth and maintenance. Then the word protein comes from the Greek word proteos, means primary, means we have to give priority, primary importance for protein because this is very essential for our body. Then carbohydrate is not essential. Yeah, it is also essential, but protein have much importance. That's all. Okay. Then amino acids. See, proteins means it is also a polymer. So what is the individual unit? So we discussed about different types of carbohydrates, but in case of protein, proteins means the monomer of protein is called a amino acid. Means proteins are made up of amino acids. Okay, so in the word itself we see amino acid means amino group should be there. Amino group should be there and acid group should be there, right? That is amino acid. So amino group means NH2 group should be there. Acid group means COOH group should be there. So the functional groups carboxyl and amino are found in amino acids. Okay. Amino acids are classified as alpha, beta, gamma and delta on the basis of position of the amino group with respect to carboxyl. See, so amino acid means I told you they contains both the groups. Amino groups are there, then acid group is also there. So based on the position of this amino group from the acid group, we can classify them into alpha, beta, gamma, delta. Important. Okay, if the amino group is means alpha position, what is alpha, beta, gamma, delta? Alpha means this one I described about this one, right? Alpha means the nearest position. Beta means the next nearest position. Okay, gamma means the next one, delta means the next one. Okay, so if the NH2 group is near to the carboxylic group, that is called a amino group, alpha group, alpha position. Okay, so glycine is an example for alpha amino acid. So why this amino acid have importance? Because proteins are made up of amino acid. Means proteins are polymers of amino acids. Okay, yes, which is sweet to taste while cheese has tyrosine. This is the structure. Okay, yes. Classification of amino acids, again important. See, amino acids are monomers of protein. So we can classify the amino acids into different types. Neutral amino acids have an equal number of amino and carboxyl group. I told you they contains amino NH2 group and COOH group, right? So neutral amino acid have equal number of amino acid. If one amino group is there, one acid group should be there. Like that. Means neutral. Means amino group is basic, right? Amino group is basic, then carboxyl group is acidic. If an amino acid contains one amino group and one carboxylic group, means it should be neutralized. How? Acid, one group, one acid, one base, means should be neutralized. So that is called a neutral amino acid. Means the equal number of acid and amino group, means basic and acidic group. Then what will be basic amino acid? Amino group is basic, I told you. Carboxyl group is acidic. So what will be basic amino acid? There will be more basic group, more amino group than that of the acid group. So basic amino acid means there should be more amino group. than that of the carboxylic group. Okay, then what will be acidic? More acidic group will be there. Okay, then non-essential amino acids are synthesized in the body, while essential amino acids are not produced. Then, see, based on these number of groups, we can classify them into neutral, basic, acidic. Okay, the first classification. Next one, based on the source, we can classify them into non-essential and non-essential and essential amino acid. Then what is the difference between non-essential and essential amino acid? Non-essential means it is obtaining, no need to get it from the diet because it is synthesizing in our body itself. So non-essential means it doesn't mean it is not important for our body. It is not like that. Non-essential means it is not, we are not getting this from external source. Okay, it is synthesizing in our body itself. Then what will be essential? This is important because it is, we have to get it from some external source. We are not synthesizing in our body. It should be getting, we have to get it from some external source, from diet. Okay, then amino acids are soluble in water and are colorless crystalline solids. Okay, amino acids act like salt due to the presence of both carboxyl and amino group. Because acyl group is there, basic group is there. So they will act as a salt. Salt means positive charge and negative charge will be there. Okay, see, sweeter ion, that is a sweeter ion actually. So an aqueous solution of amino acid become dipolar as carboxyl groups have the tendency to lose a proton while amino groups can accept a proton. See, what is a sweeter ion? Amino acid, this is an amino acid, right? Why? Because amino group is there, acid group is there. So this is an amino acid. Okay, because this is a neutral amino acid because one acidic group is there, one basic group is there. Amino acid can be exist as sweeter ionic form. What do you mean by this sweeter ionic form? Amino acids can be exist in sweeter ionic form. What is sweeter ion? Sweeter ion means, see, there is a positive and negative charge. In this one, see, here you can see positive charge here, negative charge here. This is amino acid normally. So amino acids can be exist in sweeter ion form means how this sweeter ion is forming. See, in the amino acid is COOH and NH2. That's all. Sweeter ionic form, this is COO minus and NH3 plus. Means this hydrogen, I will draw here. See, normally this is the compound RCH NH2. There is a lone pair of electron on nitrogen as usual. C double bond O OH. This is normal amino acid, right? Amino acid. How they are existing in sweeter ionic form? R CH C double bond O minus NH3 plus. How this form is converting into this? See, there is a lone pair of electron on nitrogen. So what will happen? Here there is a bond between oxygen and hydrogen. This hydrogen will be absorbed by, they will be abstracted by this NH2 group using this lone pair of electron. So this hydrogen will be released as H plus ion. Means they are not taking this bonded electron. They are losing that bonded electron. So this oxygen will get a negative charge and this hydrogen will be removed as H plus. And this H plus will abstracted by NH2 group using the lone pair. Means this NH2 group will form one bond with the H plus ion using this pair of electron. become NH3 plus. Got it? Now this is called a sweeter ion. How understood how they are converting into sweeter ionic form? Okay. The hydrogen is removing as H plus ion and this pair of electrons are remaining on the oxygen atom. Then it will be a negative charge. Then that H plus ions are abstracted by NH2 group by using the lone pair of electrons. So they will convert into NH3 plus. Why this plus charge here? Because nitrogen is using one pair of electron to make bond, right? So there is a positive charge. Structure of protein. What is the structure of protein? See, protein importance we know, the classification of protein we know. Now we are moving to the structure of protein. What is the different possible structures in which a protein can exist? Okay, see, proteins are made of amino acids which are joined by peptide bond. See, what is the difference between the carbohydrate and protein then? Carbohydrates are existing in between the monosaccharide molecules. There is a glycosidic linkage, I told you. But in protein, between the amino acid molecule, there is a peptide bond or peptide linkage. Peptide linkage or peptide bond. Okay, means actually that is an amide bond. Means CONH linkage. Okay, proteins are made of amino acids which are joined by peptide bond or peptide linkage. An amide formed between carboxyl and amino groups is a peptide linkage. So in protein, the monomers means the amino acids are linked together by peptide bond. The reaction between two molecules of identical different amino acid is initiated by the interaction of one molecule's amino group with the carboxylic group of the other one. How they are forming? See, suppose I am taking one amino acid here. NH2, CH2, COOH. Reacting with another same or different. Suppose I am writing the same thing. So it will be same molecule I am taking. Suppose this is a homopolymer, like same monomers are there, right? Yes. So CH2, COOH. Then what will happen? One water molecule will be eliminated. You remember esterification? When an acid is reacting with an alcohol, 1 mole of water molecule is removing. So the bond is forming, means COO linkage, ester linkage. Okay, same thing is here. But here, water molecule is eliminating. But this is not COO linkage. But this is NH2. So what will be the linkage? This here there is no change. CH2 no change. C double bond O no change. But this OH group is removed. So this CO group will connect to this nitrogen. So from this NH2, only one hydrogen is removing as with this OH group for removal of water molecule. This OH and one hydrogen from this NH2 is combining together to produce H2O. Because 2 hydrogen is needed for one water molecule, right? Yes. So the remaining is CO after this OH is not there. Then NH. Then the remaining thing is same. CH2, COOH. Got it? So this is the CONH linkage. This CONH linkage is called a peptide linkage. Got it? Understood? NH2, CH2, CO, NH, CH2, COOH, right? So this CONH linkage formed by the removal of one water molecule is called a peptide linkage or peptide bond, or this is also called a amide bond or amide linkage. Okay, see, removal of water molecule and the creation of peptide link CONH linkage. This is composed of 2 amino acids. I told you. Okay. Yes. So when the carboxyl, this is another example. This is the example I wrote is different. This one is different. Okay. So this one, see, amino group of alanine when produced, glycylalanine. This is glycine, this is alanine. I took 2 glycine molecules. Okay. Yes. Next. See, when a third amino acid is added to dipeptide, this results a tripeptide. See, if the protein contains 2 amino acid, that is called a dipeptide. If they contain 3, that is called a tripeptide. Okay, this contains 6, it will be hexapeptide. 5, it will be pentapeptide, like that. Got it? Based on the number of monomers, we can change the name. Di, tri, then tripeptide, then tetrapeptide, pentapeptide, hexapeptide, like that. Depends on the number of monomeric units present. Okay? When the number of such amino acids exceeds 10, the products are referred to as polypeptides. Same thing. When we are using the term poly, more than 10, right? Oh, normally we are using it is more than 2, 3, we can use the term poly. But here we are using it is more than 10, we will use polypeptide. Otherwise it will be dipeptide, tripeptide, then tetra, penta, hexa, hepta, octa, like that. Okay. A protein is a polypeptide with more than 100 amino acid residues and a molecular mass greater than 10,000 units. Means they contain large number of monomers. So polypeptide means more than 100 will be there. So the molecular mass will also increases. When the number of the individual units are increasing, the molecular mass will also increases. Got it? Yes. Next one is classification of proteins. Okay. So we discussed about the structure of protein. Then proteins are made up of amino acids, I told you. Then between the amino acid molecule, there is a linkage called a peptide linkage. How the peptide linkage is forming? By the removal of water molecule. Okay. Then the proteins can also exist in sweeter ionic form, I told you. Okay. Next one is classification. So the polymers can be classified into different way. The important classification, I told you, because the protein is a polymer, so the protein can be classified on the basis of different amino acids be classified, essential, non-essential, like that. So proteins can be mainly classified into 2, that is called fibrous proteins and globular proteins. Fibrous proteins and globular proteins. So what do you mean by fibrous proteins? A fiber-like structure is generated when the polypeptide chains run parallel and are kept together by hydrogen and disulfide bonds. Okay, there is a fiber-like structure. Fiber-like structure like this. Okay. Then when polypeptide chains are run parallel and are kept together by hydrogen and disulfide bonds. Like this. Hydrogen and disulfide bond. This bond between 2 different chains will be hydrogen and disulfide bond. Hydrogen bond we know, right? So disulfide bond is also there. So between the 2 fibrous chains, the bond is there because there should be a connection between the 2 chains, right? The connection between the 2 chain is actually hydrogen bond or disulfide bond. These proteins are not soluble in water. They are insoluble in water. Example is myosin, found in muscles, and keratin, which is found in hair, wool, and silk. Okay. So what are the examples for fibrous protein? Keratin is there. Then which one? Myosin is there. Okay. Yes. Next one is globular proteins. This structure is formed when spherical polypeptide chain coil around one another. Globular. Globular means it is not like a straight chain. Fibrous protein means it should be a straight line like this. Two lines are running parallelly and they are connected by hydrogen bond. Globular means it is a coiled structure. Okay, globular protein. There will be granules like that. Okay, then these are water soluble. See, fibrous proteins are insoluble in water, but globular proteins are soluble in water. Albumin and insulin are two common globular proteins. So, fibrous protein examples means keratin, we can say myosin, right? Globular protein means the important albumin, insulin, they are globular proteins. Protein structure and shape can be investigated at 4 separate levels: primary, secondary, tertiary, quaternary, with each level becoming more complex than the previous one. Again, important. See, in the board exams, you can see the questions from the same topic. Explain about the primary, secondary, tertiary and quaternary structure of protein. We can see the same question in your past papers. Okay, then what is this primary, secondary, tertiary structures of proteins? We know primary carbocation, secondary, tertiary, that all we know, right? This is different. This is different from that of that organic chemistry, like the primary structure, the carbon is connected to one algae. Not like that. This is different. Got it? Yes. So, fibrous protein, I told you, two fibers are running parallelly connected by hydrogen bond. Globular protein means they will be coiled like this. Okay. Then, in case of globular proteins, we can again classify them into different based on their structure. So, they are primary structure, secondary structure, tertiary structure, quaternary structure. But see, when you are moving from primary to secondary, secondary to tertiary, tertiary to means this become more complicated. I will show you how they are forming these different structures. Okay, then first one, see, this is the primary structure. I will go to the previous slide. Before that, I am explaining. See, this is the primary structure. See, all these individuals, you can see, right? I will zoom it. See, here you see these individual beads like structure, they are amino acids. They are amino acids. Because the proteins are, means the protein chain is made up of different amino acids. It's a polymer, right? So these colored beads like things are amino acids. So as I told you, one chain may contain different types of monomers, right? One polymer, one protein chain may contain maybe same monomer will be there, or different monomers will be there. So these different colors means they are made up of different types of monomers. Okay, see, so this is primary structure, means they are straight chain like this. See, primary. Secondary means this primary structure will coil themselves right like this. See, they are like helical, means they will coil themselves like this and they will form the secondary structures. Then what is tertiary? Tertiary means, see, it become again complicated. Like, see, these two, suppose two secondary chains are joined together. It is like that, right? See, this is a secondary structure. This is another one. So when these two secondary chains are joined together, you will get this tertiary. Means it become more coiled, it become more complicated when you are moving from secondary to tertiary. Okay, then what is quaternary? Again it become, see, again they will self-coil together to form this quaternary structure. It is always not like this one, this one, this one, then it is, no, this is an example only. They can coil in any way. But see, primary is a straight one, secondary, that one is self-coiling, tertiary means two different secondary are coiling themselves, means it is not that much complicated. Tertiary is not that much complicated, but quaternary means it is very complicated. See the structure. Got it. This is actually the primary, secondary and tertiary structures. Okay. So before that, we have to discuss about it. See, so the structure is clear, right? Otherwise, if I am explaining only the theories, you won't understand. That's why I showed you this one, the structure first. So the structure is clear, right? Yes. So we are moving to the explanation. See, primary structure. I told you primary means this one, only single chain made up of many amino acid. In proteins, one or more polypeptide chain can be found. We know that. Each polypeptide in a protein has amino acid linked together in a precise sequence, and the sequence of amino acid is referred to as protein's fundamental structure. This is as I told you. See, this is the same thing I told you. What is that? See, a polymer means a protein is made up of different types of amino acid. The amino acid used to synthesize this protein may be same or different. Okay, so in protein, one or more polypeptide chains can be found. Each polypeptide is made up of maybe same or different. That sequence is determining the structure of that protein. We have different types of protein, I told you, right? So how these all different types of polymers are different, how all these type of different proteins are varying together because of this sequence of these monomers. Suppose one example, in one protein you can see the sequence AB, ABC, AB, ABC. In another protein it may be BACD, BACD. Got it? Understood the difference, right? Maybe the monomers may vary and the alignment, see the sequence may be also, they also may vary. Then the complete structure, functions, everything will change. Whenever the sequence and the individual units of the proteins are changing, the complete functions will also change. That's why different proteins have different functions in our body. Even one unit is changing, entire properties will change. The properties of this one may be different, entirely different from that of the second one. Okay, so a protein is produced when it's this fundamental structure or the amino acid sequence is altered. This one, as I told you, the first structure, this one. Okay. Next is secondary structure. What is secondary? The main means the primary structure is coiling themselves like this, right? This is secondary. This is primary. This is secondary. Secondary structure is the configuration in which a lengthy polypeptide chain can exist. They have been discovered to exist in two different structures, alpha helix and beta pleated sheet. So primary, secondary, tertiary, quaternary is there. In the secondary itself, there is alpha helix structure and beta pleated sheet. Okay? Yes. Then, see this one. See the difference? Okay? Got it. Then, tertiary structure. Protein tertiary structure represents total folding of polypeptide chain, that is folding beyond the secondary structure. What is that? See, this is one secondary structure. This is another secondary structure. As I told you, two secondary structures are, means again they are combining together. It becomes more complicated than that of the secondary structure. Okay. So it produces two major molecular shades, globular and fibrous. This one already we discussed about it, right? Globular and fibrous. In globular, two parallel structures will be there connected by hydrogen bond. Then this is fibrous. Globular, coiled one. Okay, this is fibrous. This is globular. Okay. Hydrogen bonds means this in fibrous, the individual means the two parallel the fiber you can see, two parallel chains you can see, they are connected by hydrogen bond or disulfide bond. Okay, van der Waals force, electrostatic force of attraction are principal forces that keeps secondary and tertiary architecture stable. Okay, then the last one is quaternary structure. Two or more polypeptide chains referred to as subunits means make up of some proteins. The spatial arrangement of the subunit relation to, see the same thing. Quaternary means it become again more complicated like this. See, it become like this. Anyway, they can coil together. So it will be quaternary structures. Okay, quaternary structure. Got it. Again important. What do you mean by denaturation of protein? Okay, denaturation. Hydrogen bonds are Rupture when a protein in its natural state is exposed to physical change like temperature change or chemical change like pH change. As a result, globules unfold, helices uncoil, and protein loses biological function. This is known as protein denaturation. Simply, we can say denaturation of protein means they are losing their biological function. They are losing their biological function. Important. In board exams, previous past papers, you can see the repeated question is from this one, denaturation. Losing their biological function or using their biological activity. I told you, depends on the sequence of the different amino acids used for making the protein. Okay, depends on the types of amino acid used for making the protein chain. Their function, structures, everything will change, right? I told you. So, if something is happened on the protein molecule, which will change its natural state, means suppose some change, suppose the protein, if we increase the temperature, if we increase the pH, suppose a temperature change, atmospheric temperature of means wherever the protein is existing, the temperature is changed or the pH of the medium is changed. Then what will happen? It will affect the chain structure of the protein. Sometimes it will cause the means the sequence of these amino acids will change. A, B, C, D become C, D, B, A. Okay. Sometimes what will happen? They may be replaced. Suppose A, B, C, D, C is replaced by some amino acid P. Got it? So these type of changes will take place when these protein molecule is exposed to some physical change. What do you mean by physical change? Like temperature change, pH change, like that. Or some other chemical change, as I told you, because this changing the sequence like that. So the entire properties of the proteins will change. The entire function. Suppose a protein is for doing a function, particular function, when the sequence is changed, the protein is not able to do that function. It means the protein is undergoing denaturation. Got it? Means due to these changes, due to these some physical changes, the proteins are losing their biological activity due to their structure, change in the structure. Due to because they become unfold. Unfold means, see, all the polymers are this one. I told you, because suppose they are like this, they are folded. Okay, so what will happen? Some changes will cause the unfolding of this chain, and proteins lose their biological function is known as protein denaturation. Okay, so denaturation destroys secondary and tertiary structure because sometimes they will cause unfolding. Secondary means this one, right? So this polymer is responsible for some function. Suppose a pH change is changing the structure into like this. Means some particular change in the pH, they are changing the structure of this protein into this unfolded manner. It means this protein, this is for a particular function A. If the pH is changed, the structure is changed, right? Now they are not able to do the function A. They will do another function B. This may be harmful to our body. Then they will affect our body. Got it? Yes. A common example for denaturation is the coagulation of egg white when heated and milk curding, which is caused by the bacteria found in milk producing lactic acid. So these are the examples. Coagulation of egg white. Okay, so these are the example for denaturation, because coagulation of egg white means their complete properties are changing. Okay, then change in the milk. That is also some changes are, some chemical changes are happening in the entire properties will change, right? Yes. So this is called a denaturation of protein. Okay. The functions will change, the sequence will change, the properties will change. Sometimes they may become harmful to our body also. Got it? Yes. That is denaturation.